Welcome to Nikolai's Genetics Lessons, and today's question: did I shear my DNA by vortexing colony PCR product I got from one of my subscribers? And I highly recommend you to watch this video because this is classical mistake which every other student can make during gel electrophoresis. So I recommend you to watch 5-minute video instead of spending hours redoing your experiment. So here's the background. I conducted PCR on several grids plate isolations to amplify the DNA for gel electrophoresis. Before running the gel, I noticed sediment or precipitant in my PCR tubes. So let me highlight these words, and we are talking about this precipitate. This is how your DNA sample usually looks like after centrifugation. I thought that I needed to resuspend the DNA. That is correct, that's true. I vortexed 4 out of 5 samples in ascending order. So this is going to be number 1, 2, 3, 4, and 5. So these 4 out of 5 were vortexed. From well sample 5 to sample 2, from 5 to 2, and sample 1 was not vortexed, so this one was not vortexed. Next, I was made aware that vortexing was not recommended, so I stopped. I did try to plug and aspirate sample 1. I used ladder here and filled 5 wells of the gel. The gel ran for 13 minutes and I got a lot of migration and smearing in the wells 3 to 5. Actually from 2 to 5. And migration in well 3, 5 exceeded the ladder. So ladder, as you see, ends here, and this is where we see the front. I sheared my DNA. I didn't observe any bending or migration in well number 1. First of all, this person sometimes start counting from this well as a 1, and sometimes he refers this well as number 1. So just to avoid a confusion, I just number them. And actually this is not true. The last statement, we do see here a bend. Those not as bright as in the other wells. Now let's analyze what we see on this picture. Smearing usually suggests from well 2 to 5 degraded or sheared DNA, which can occur due to excessive handling such as vortexing. Vortexing can shear long DNA molecules, leading to smaller fragments that can create smeared appearance on the gel, what you see here, and the front also going to be made by DNA which is smaller than supposed to be, and this would travel even farther than the ladder itself. Since, as you see, this person vortex samples 2 to 5, this likely caused the observed smearing and excessive migration, especially if the migration exceeded the ladder, which indicates fragments of varying sizes. Now let's analyze what is in the well number 1, why it's different from the rest. If you do not vortex, but you still have some DNA in suspension form, you draw it with a pipette, but a lot of DNA is not going to be drawn in the pipette, and that explains this faint bend. If all DNA would precipitate and you're going to draw this liquid here, you're probably not even going to get such faint bend at all. So how to avoid this common mistake? I would recommend to avoid vortexing the PCR product and instead use gentle pipetting to mix. Gentle pipetting is not going to shear your DNA sample, and you're going to get everything in suspension form, and you're going to get nice bend in the proper place because DNA is not going to be sheared by vortexing. And this is all for today. Subscribe and see you in the next video. Goodbye.